All right, welcome back, guys. Um, so over the last several lectures, we've been talking about simple chemical kinetics, right? Um, and I've pointed out how a really important aspect of understanding chemical kinetics is this idea of transition state theory, right? This idea that the rate constants we talk about when we think about chemical kinetics are directly related to this transition state free energy barrier that any chemical reaction has to cross, going from the reactant state to the product state, right? Um, and the additional thing I pointed out here is one of the reasons transition state theory is so useful, in addition to linking up the value of these rate constants to this free energy barrier between, say, the product state and the transition state, is because this also gives us this simple way to understand catalysis, right? Where catalysis is simply a matter of making that free energy barrier smaller, right, compared to the uncatalyzed reaction, right? And that this is what enzymes are doing, right? And we spent some time looking at some examples and sort of descriptively about how enzymes could do that, these sort of 6 different types of catalysis, acid base, covalent, et cetera, through proximity and orientation, and just general transition state stabilization. And that idea of transition state stabilization is sort of the big idea here, right? You know, stabilizing the transition state for a reaction is sort of part and parcel of what enzymes do to make reactions get to equilibrium faster, to catalyze chemical reactions. Okay, so that's great, but we need to take the next step. We want to get quantitative about this, right? We want to understand something about these enzyme mechanisms in quantitative detail, right? This is sort of where we're headed. And you could sort of then fairly ask the question, well, why? Why is it so important to understand these mechanisms, right? And there are lots of different reasons. And a preliminary step in getting to the kind of information, or an essential step to getting to some of the information, like we sort of outlined last time, when we looked at the mechanism of the serine proteases, involves understanding the temporal progression of these reactions, so-called enzyme kinetics. You can sort of imagine this is where we were going, given we started with chemical kinetics, sort of the bigger picture. And now we're going to bring those ideas together to understanding enzyme function. But it's still worth asking the question, well, why? Why is understanding enzyme kinetics, and therefore as a tool for understanding mechanisms, such a big deal? Well, for one thing, consider the phenomenon that chemists who identified some of the first enzymes faced when they started looking at how these remarkable biological catalysts seem to be working. When they took a look at the chemical reaction, the velocity of the chemical reaction, these enzymes, these amazing little catalysts were doing as a function of, say, the substrate molecule, what they saw is kinetic behavior that didn't look an awful lot like simple chemical reactions. Instead, these reactions did something like this, right? You imagine here our data points here, right? And so you think about, from the point of view of people used to chemical kinetics, how on earth do you explain something like that? That doesn't look like any normal chemical reaction, simple chemical reaction. So how do you explain that kind of behavior? So, you know, there's a good reason for wanting to understand enzyme kinetics, right? Just the pure reason of understanding something about the universe that is fundamental. How does that work? Why does it look like that, right? In addition, another good reason, like I mentioned before, is a lot of those mechanisms that we sort of outlined, many aspects of those mechanisms were elucidated, or can be elucidated, by thinking about enzyme kinetics. And enzyme kinetics is a broad term. It applies to just about any of the ways in which one would study the temporal behavior of a chemical reaction that is catalyzed by an enzyme. We're going to spend some time dealing with one particular stripe of that sort of analysis, right? And then there's a third and really practical reason for trying to understand the enzyme kinetics, right? And that has everything in the world to do with drug design, right? So a huge number of the therapeutics and the small molecules and the drugs we associate with modulating or curing different types of disorders or diseases are designed to target or modulate or inhibit or modify in some way specific enzymes, right? Huge fraction of them, not all, but lots and lots of the important therapeutics actually target and modify the activity of enzymes, right? And not covalently, right? They modify their chemical properties, right? So as a specific example of that, consider, right, the way in which HIV, the human immunodeficiency virus, creates the problem it does when it invades a human immune cell to begin wreaking its havoc, right? So when HIV invades, it generates essentially all of its proteins initially as two very large polyproteins, right? One is of course known as GAG. The other is known as GAG-PAL. And if you want to know a lot more about this, a good person to talk to in our department is Tom Meek. He's done a lot of work in this system. And there's some, there's some leader sequence stuff here, right? Some bits here, right? And a tail end piece here. GAG-PAL, sort of the same way. Whoops, there we go again. And then we have here, and there's some more intervening sequence stuff here. But the really key thing that, for my purposes right now, is a pair of proteins that are synthesized as this initial huge polyprotein. One is that really key reverse transcriptase, right? And just amino terminal to that, remember this is a polyprotein, is the so-called HIV protease, right? Now, if these polyproteins and these associated open reading frames are not excised from this polyprotein, the virus is dead. There's no more replication. The reverse transcriptase has to be replicated to make copies, and you gotta make some of these other proteins in order to synthesize the associated tegument proteins and stuff that go into making HIV viral particle. And all of that depends upon the activity of this protein, the HIV protease, and its ability to excise itself and then go around and chop up these polyproteins into their associated activities. And if it can't do that, the virus is dead. Okay, so the enzymatic activity of the HIV protease more or less sits as an Achilles heel for this virus in some ways. If you could take out, in principle, if you could take out the HIV protease, if you could prevent it from doing that, you could shut down an HIV infection in principle, right? And turns out that that activity, targeting that activity, has been a hugely important aspect of HIV therapeutics. And in order to develop the approaches to knowing if a given molecule is good, if it's not good, can we make it better, that utterly depends upon characterizing the enzymatic activity of the HIV protease in the presence and absence of different small molecules, right? And that, at least in a first pass, depends upon enzyme kinetics, right? So if you want a good practical reason for why enzyme kinetics is so important, here's one right here. All right, so we're going to dig into how enzyme kinetics works in the next video.